Despite its complexity, the organs of the human body are composed of four basic tissue types: epithelium, connective tissue, muscle, and nervous tissue. Although the concept of tissues comes from work done in the 1800s, it has stood the test of time and still provides a useful way of understanding normal function and recognizing disease. This video will discuss epithelia. Before we get to epithelial tissues, here are a couple of definitions that will be useful as we think about tissues and organs throughout the unit. Organs are divided into parenchyma, which consists of the cells that are responsible for that organ's function, and stroma, which is essentially everything else in the organ. Here are a couple of examples of parenchyma and stroma for different organs. The parenchyma of the heart, for example, is muscle tissue, while the nerves, blood vessels, and connective tissue comprise the stroma. Now, there are 9 different types of epithelia, but we will not cover those in this video. You can access your learning module for Histology Lab or read about the 9 types in the Big Picture Histology, Chapter 1. First, we need to understand what are epithelial tissues and why are they important. Epithelia line all external and internal surfaces of the body, and everything that enters or leaves an organ must cross some type of epithelium. The main functions of epithelial tissues are protection, absorption, and secretion. And since these are often at a free surface, epithelia are specialized to hold on and stay intact. We'll talk about how that works shortly. Also, being at a surface means that one of the major roles of epithelia is protection. Most epithelia provide a mechanical barrier, and in some cases a chemical one. Here are 2 examples of chemical barriers: the mucus secreted by respiratory epithelium, and on the right, defensins produced by cells of the small intestine. Here we have some of the major characteristics of epithelial cells. You should know these, but note that we will discuss glands later in the unit. 2 important characteristics of epithelial tissue are its ability to divide, which is important for wound healing, and its lack of blood vessels. In addition, you will see that because of their location, epithelia have specializations that enable them to perform their functions, and we'll discuss some of these specializations in a few minutes. Importantly, epithelia show polarity, with an apical surface that faces the space or lumen, which you might remember is effectively outside the body, as in the lumen of the digestive tract where food or chyme moves along. The basal pole faces inside and contacts the connective tissue. Organelles and proteins are distributed unevenly inside the cells, which compartmentalizes their functions. The 2 regions thus differ in structure and in function. There are important junctions that help maintain this polarity, which is required for many of the functions of epithelial tissues. Now, the apical ends of many epithelial cells have specialized structures that protrude from the cells, and these function either to increase surface area, as in the intestines, or move substances along, such as in the respiratory tract. These structures include microvilli, stereocilia, and cilia, and we'll discuss each of these as we move through the different organ systems later in the unit. Let's take a closer look at the basal pole. The basal surface of all epithelia rests on a thin extracellular sheet of macromolecules known as the basement membrane. The basement membrane is a semi-permeable filter that controls substances reaching epithelial cells from below. Other functions of this membrane include providing structural support for epithelial cells, attaching these cells to the underlying connective tissue, organizing and maintaining polarity, and helping localize many different activities of the cells. Basement membranes are also critical for cell-to-cell interactions, cell migrations, and epithelial repair and regeneration. Thus, disruptions of the basement membrane can have serious consequences for epithelial tissue and organ function. And we'll see some of these consequences in lab and in our team-based learning exercises. As we see here in this H&E stain section and in the cartoon, the basement membrane appears as a thin line that is difficult to resolve under the light microscope. Both epithelium and connective tissue contribute to the composition of the basement membrane. Now, this transmission electron micrograph shows the basement membrane a bit more clearly. Here you can see a network of fine fibrils that comprises the basal lamina, shown by the arrow, and beneath that is a fibrous reticular lamina or layer. The components of basal lamina include collagen and laminin, connective tissue components that we will describe when we study connective tissue. We often use the terms basal lamina and basement membrane somewhat interchangeably, but basal lamina is more strictly an EM term, while basement membrane refers more to the entire structure beneath the epithelial cells under the light microscope. As I mentioned earlier, epithelial cells contain specialized membrane-associated structures that provide communication and adhesion between cells. As you might imagine, epithelial cells, particularly those subject to friction or mechanical forces, such as in the skin or oral mucosa, adhere quite strongly to neighboring cells and to the basement membrane. This cartoon summarizes the different types of membrane-associated structures, and we'll now walk through these one by one, starting with the junctions that are located on the apical lateral surfaces of the cells. The first intracellular junction we'll talk about is the tight or occluding junction, also often called zona or zonula occludens. One of the things you'll have to get used to in histology is that structures often have many different names. Some of these are historical, while some refer to the terms that a pathologist would use. Zonula indicates the junction forms a band completely encircling the cells. In transmission electron microscopy, the membrane will actually appear fused. Tight junctions form seals between adjacent cells and serve to prevent passive flow of material between the cells. In other words, the tight junction ensures molecules that cross an epithelium do so by going through cells and not between them. So an epithelium that needs to be leak-proof, such as the bladder, will generally contain more tight junctions than a cell that requires permeability to function, such as the kidney. There are specific proteins associated with each type of junction. Here we have occludens and zona occludens proteins, or ZO proteins. Mutations or disruptions of these proteins can lead to disease. For example, food poisoning can be caused by bacteria that target tight junctions, interrupting the function of the junctions or the formation of the junctions, causing a loss of tissue fluid into the lumen of the gut and eventually diarrhea. Adherens junctions, or zonula adherens, are typically found close to tight junctions, and like tight junctions form a continuous band around the cell. They are typically just basal to the tight junctions and serve to stabilize and strengthen the occluding bands. They help to hold the cells together. Together, the tight junctions and adherent junctions act similarly to those plastic rings that hold 6 packs of Diet Pepsi together. In adherens junctions, the main protein components are members of the cadherin family, which link to actin filaments. And loss of cadherins can lead to loss of cell-to-cell adhesion. Another anchoring junction is the desmosome or macula adherens. Unlike the tight junction and adherens junction, desmosomes don't encircle the entire cell. This junction is more like a single spot weld. Desmosomes are structures at the surface of one cell that are matched with identical structures on the adjacent cell surface. Cell adhesion by desmosomes is mediated by large members of the cadherin family called desmoglines and desmocollins. Catenin-like proteins called plakoglobins and desmoplakins also link to intermediate filaments rather than actins. These filaments are called tonofilaments and contain different keratins. What I need you to remember here are desmoglines and keratins. Blistering diseases often arise from abnormal desmosome function. Gap junctions are a type of communications junction. So rather than adhering or occluding, they mediate intracellular communication. These junctions are actually complexes of transmembrane proteins that form patches in the plasma membrane. Connexin proteins form channels that permit intracellular exchange of small molecules, and this exchange can allow cells to act in a coordinated manner, such as in heart contraction. On the basal epithelial surface, cells attach to the basal lamina by anchoring junctions called hemidesmosomes, which can be seen through transmission electron microscopy. Ultrastructurally, these resemble a half desmosome, but the proteins in hemidesmosomes are called integrins, and they directly bind to laminins, a connective tissue component, and indirectly to intermediate filaments in the basal lamina. A related basal anchoring junction is the focal adhesion, in which integrins link directly to actin filaments rather than intermediate filaments. Now, all cells need to control ion and water transport, in either direction, a process called transcellular transport. So let's review some of the basics of transport. There are multiple ways that molecules pass membranes. Oxygen and carbon dioxide are capable of simple diffusion, while some molecules pass through channels or need to be brought in via a carrier or actively transported, and some of these processes require energy. Control of ion and water movement is a critical function of epithelial tissues, and misregulation of this process can have serious consequences. We will examine transport through our unit as we move through the different organ systems, but for now, here's a simplified diagram of an intestinal epithelial cell showing some of the important transporters, channels, and junctions. Let's walk through this together. All cells can internalize extracellular molecules and fluids using endocytosis and formation of membrane-bound vesicles, and this process is termed transcytosis. But epithelial tissues need to actually be able to actively transport ions against concentration and against electrical gradients. A good example is the sodium-potassium ATPase or sodium-potassium pump, which allows cells to maintain a required low intracellular sodium and a large excess of potassium ions on the inside of the cell. This is an active transport process involving the hydrolysis of ATP to provide energy. So as I mentioned, epithelial cells also have to control the passage of water, and this requires even more junctions. So transport functions require exquisite control and compartmentalization, and that role is in part performed by tight junctions. On the right here, you will see a demonstration of how tight junctions prevent the passage of tracer. You can see how the tracer remains confined to the apical compartment. Now, as alluded to earlier when we talked about tight junctions, water can leak through epithelial cells in a process called paracellular transport, which is controlled in part through these tight junctions. And normally, water movement here is quite low. Cells also contain membrane channels that allow the passage of water, termed aquaporins. Aquaporins are shown here in gray, and they allow the passive transport of water by osmosis. The direction that water flows is determined by the relative solute concentrations. Osmotic and electrical balance is maintained by different ion channels. Here we have two examples, a chloride and a sodium channel, shown here in pink and yellow respectively. One important chloride channel that you will encounter many times in this unit is the cystic fibrosis transmembrane conductance regulator, or CFTR channel. In part, this channel also functions to control the ENaC sodium channel. Mutations in the gene that encodes CFTR can lead to imbalances in the chloride and sodium ion movements, and as a result, changes in water transport. And the consequences of the mutation depend on the type of mutation and the particular role of the CFTR channel in a specific organ. For example, in the digestive system, mutations in CFTR can lead to too much export of water, leading to diarrhea. However, in the respiratory system, mutations in CFTR can have a different effect. If there's defective protein, the amount of secreted chloride is decreased. Subsequently, there is an increase in the amount of sodium transport into the cell. Now, due to these disruptions in the gradient, water will follow the sodium, decreasing the amount in the lumen of the respiratory vessels. Thus, that leads to thick mucus that can trap bacteria, increase obstruction, and increase infections. Not surprisingly, epithelial tissues are subject to damage. Their function is protection, so they're subject to both mechanical and chemical damage. Because of this, epithelial tissues are relatively labile structures, and their cells undergo continuous renewal. This occurs by mitotic activity and involves activation of stem cell populations. The rate of renewal of an epithelium varies with the type of epithelium. So intestinal epithelium has a fairly fast renewal rate, while glandular epithelium renews more slowly. The locations of stem cell niches also vary among different epithelium. Stem cells in stratified epithelium, such as skin, are located only within the basal layer that is in contact with the basal lamina. But in contrast, stem cells in the intestine are located in restricted niches that are some distance away from differentiating cells. In this example from the small intestine, Stem cells are found in a specific niche called a crypt that is located at the base of intestinal villi. The renewal process here is complex and controlled by a genetic cascade shown here on the right, which is constantly being updated. Now, interestingly, although the steps may vary, many of the same genes and signaling proteins control epithelial turnover in many different epithelia. So not surprisingly, mutations in genes in these pathways are often mutated in disease, and particularly in cancers. For example, uncontrolled expression of Wnt signaling is associated with colon cancer. I don't expect you to memorize this pathway, but you should be familiar with major players such as Wnt's and BMPs, because they are often targets for chemotherapy or other interventions, and you'll hear about them again in this unit. In addition to self-renewal, epithelial cells can also undergo transformations. One type of epithelial tissue may undergo transformation into another type in a process called metaplasia. Shown here on the right is metaplasia of columnar epithelium to stratified squamous epithelium. This process could be protective, as is thought for the case of metaplasia of esophageal tissue that occurs with acid reflux, where the cells transform in order to protect themselves against that acid. In metaplasia, cells of one mature differentiated type are replaced by cells of another mature differentiated type. Metaplasia is often reversible when the stimulus is removed in particular. Abnormal growth or differentiation of epithelial tissues is called dysplasia. Usually, dysplasia refers to expansion of immature cells at the expense of mature cells. Sometimes these changes are considered precancerous, and they can be termed neoplasias. Dysplasias can be considered precancerous lesions, and the degree of dysplasia often can correlate with the risk of cancer. In this cartoon and micrograph shown on the left, in the top panel we have the cartoon showing progressive stages in the development of malignant carcinoma, which is then mirrored in the H&E sections below. The first dysplastic changes are seen in the basal cell layer of this stratified squamous epithelium. That's the layer closest to the basal lamina. Now note that there isn't a transformation into a different epithelial type as we saw on the previous slide, but instead this is abnormal growth of the basal cells. The next step, carcinoma in situ, represents the transformation of a neoplastic lesion to one in which the cells fail to mature, they lose their tissue identity, and begin to grow and divide rapidly without normal regulation. Finally, the cells invade past the basement membrane and now have the potential to move to other parts of the body. You'll learn a lot more about these terms from pathologists later in our unit and see several examples of dysplasia when we study the reproductive system in a few weeks. Finally, let's end with a few definitions. Cancers derived from epithelial cells, such as the one shown above, are termed carcinomas, while cancers derived from glandular epithelia are called adenocarcinomas. And we'll look at some examples in histology lab of both of these types. That's it for epithelial tissues. Don't worry if you can't master all this material at once. We'll be looking at epithelia throughout the unit, and we'll be talking about the specific structures and functions of epithelia in various organs. So there will be plenty of opportunities for reinforcement of major concepts and details.